Hello, everyone, and welcome. We're giving everyone a few seconds for joining from the waiting room. Today it's July 7th, and this is Sabrina Paganoni. We're giving our weekly update from the HEALEY Center for ALS and Mass General. This is about the HEALEY ALS Platform Trial, but as always, we want to provide you with information, important and relevant information about ALS research in general. And so tonight I'm here with our patient navigation team, as always. Alison and Catherine are here with us, and they're always available outside of this meeting to answer your questions. And if we can go to the next slide, it's really an honor for me to introduce my close colleague and friend, James Barry, who wears many hats, and in this context is the co-lead investigator of Regimen C, and also I should mention of relevance for tonight's meeting, you are the chair of the biomarker committee that's part of this trial, and also the chair of the NIS biorepository, where we store so many of the biosamples that then allow for biomarker analysis, and co-chair the NIS consortium. And I'll stop there because I know you have many titles, but really, well, what's really amazing is to work with you, and especially tonight, we invited you to tell us more about what's new and important in ALS biomarkers. And so with that, I'll turn it over to you, James.
Great, thank you. Should we go to the next? Thank you very much, Sabrina. We can go to the next slide. It's really an honor to be here. I love being a part of these webinars, and I get to step on periodically, and it's just such a great thing for the community. I thank you for having me. It's an exciting time for the platform trial, and I think a good time to reflect on what we're putting together as what we're calling exploratory endpoints, and are, I think, becoming more and more central to what we do, which is a really good and important thing. If we look at some of the advancement that's happened in oncology, cancer, over the last decade or decade and a half, a lot of that has to do with the fact that they've developed good biomarkers to diagnose and also follow progression in oncology. Now, that's not the only reason there have been successes, but that's one of the reasons. And I think our field, the ALS community, has really put a lot of importance on this over the last 10 years as well. And we're turning a corner with some biomarkers and getting them to a point where they can and should be useful in trials. And we'll talk about neurofilament, which has particularly turned that corner. But then there are other biofluid biomarkers, that is, biomarkers we look for in urine, blood, spinal fluid, also known as CSF, and even some of these digital biomarkers that are beginning to lead the way as well. I'll come to DNA at the end. We'll leave that to the very end, even though it's the first thing on the slide. So let's start with neurofilaments. Neurofilaments are the scaffolding protein of neurons, and they sort of hold the structure of the neurons. And as the neurons become ill or are lost, they spill the contents of the neurofilaments into the spinal fluid and the blood. They go into both compartments, into blood and into spinal fluid, and we can test for them there. There are actually 3 species of neurofilaments. There's heavy, medium, and light chain. We have assays for 2 of them that are mostly specific, which are light and heavy chain. And all that is really nitty-gritty and in the details, but what we're sort of settling on is that we can do either heavy chain analysis or light chain analysis of neurofilament. We're doing a little more light chain. I think our assays are a little bit better in blood for light chain, but it's nose and nose, it's sort of a photo finish. In the platform trial, we're looking at neurofilament lights. Now, here's what we know about neurofilament, is that, and we know this from many studies over many years, from many different groups, been replicated and replicated, and we feel very confident about this, is that when someone has ALS, we can measure the neurofilament, and higher neurofilament levels tend to correlate with faster disease progression. Now, this is not a perfect correlation. Like everything in ALS, there's some variability, but overall, it's a rule that holds. Neurofilament is not specific to ALS. In other words, there are other conditions where neurofilament can go up. So that's true in Alzheimer's disease. It's true in frontotemporal dementia. It's true in nerve injury. So a traumatic brain injury, for example. And even some of the kinds of neuropathies can have rises in neurofilament. So what that means is we can't do a neurofilament test and say this person has ALS or doesn't have ALS. We have to confirm the diagnosis a different way. And then we can learn something from one measurement of neurofilament. In trials, we're using that that baseline measurement, or right when people enter a study, we can measure neurofilament, and we can say, well, this person living with ALS has an elevated neurofilament, and we might categorize them as expected to be a rapid progressor, and someone else might have a much more subtle elevation, and we might categorize them as a slow progressor. And that can help us with the way that we kind of subgroup people and do our analyses, and we can actually get some sort of statistical edge from that, getting more insight from the same number of people and reducing the number of people that we need on placebo by incorporating that information. And that's really, I think that's really a benefit. We also can follow neurofilament over time. And the other thing we know from many, many studies is that neurofilament is more or less stable over time. So if we measure it in the blood today, and then we measure it in the blood 3 months from now, in a person living with ALS, that number would be very similar at those 2 time points. And if we measure at 3 time points, it'd be very similar. There's some up and down, but it'd be pretty similar. And so then we can use this not only to sort of say, well, this person may be a fast progressor and this person may be a slow progressor, but now we can actually use it as, to say, well, it should be stable in the natural history of things. Can we get, can we assign them to a study drug and see whether the neurofilament declines? And we've seen examples of this now. In the tofersen study, we see a very robust, these are results that have been presented a few times now, see a very robust reduction in neurofilament that happened in the treatment group and not the placebo group. And that's, so we can now use it as a biomarker, as an endpoint in the trial. There are still limitations to this that are really important, which is that we're really beginning to understand neurofilament. I mean, there's a lot we know about it. I've already told you a lot that we know, and that's hard won, and it represents lots of work from lots of groups over a long period of time. But there's still things we don't know for sure. Like, for example, can a treatment work clinically without reducing neurofilament? I think we can imagine ways that could be true. And so I think we're hesitant to say that something has to reduce neurofilament in order to be considered effective. I think we would largely, any of us in the field would be hesitant about that statement. Another place we're not sure about what to think is, what if there's a small increase in neurofilament with a treatment, but that treatment seems to have some clinical benefit? There's some things to work through. What if these two things diverge? We would have to think about that. I think in the end, the clinical outcomes are the ones that probably take precedent, but we think neurofilament can help guide us. We're also looking for other biomarkers in blood, urine, and spinal fluid. So spinal fluid is closest to the motor neurons. It's what bathes the brain and the spinal cord. And it flows from the brain down over the spinal cord and back up to the brain where it's reabsorbed. And it bathes the brain, it bathes the brain and the spinal cord in a sort of a conducive environment for function. It also provides some cushion. And so it has a lot of roles. Because it's in direct contact with the central nervous system, it's a source of potential biomarkers, proteins that are shed into the spinal fluid, potentially RNAs that are shed into the spinal fluid, microRNAs that are shed into the spinal fluid. These are all things that we can analyze in the spinal fluid. In order to get to spinal fluid, we have to do a lumbar puncture. That is a part of the platform trial. We can do a lumbar puncture. We have not yet required that as any of the regimens. And that's because we understand that it can be challenging to go through. At the same time, there is some really critical information that we can get from spinal fluid on the biomarker front. Some of that may be specific to a drug regimen. So there may be a drug regimen where we really are interested in what the drug levels are in the spinal fluid, because if those are too low, then we maybe are using the wrong dose. And that can be critical to know before we move forward. So there are some, that's one example, but there are some reasons that getting spinal fluid can be really, really important. We also know that there are some inflammatory markers that really we can see in the spinal fluid that we can't see in blood. There's one called MCP1, which has a much longer name, that monocyte chemoattractive protein 1. Doesn't matter. MCP1 is something that goes up in ALS. We can measure it in blood, but it doesn't give us a very clear signal in blood. It does give us potentially a clear signal in CSF, and one that we might want to follow. It doesn't, I think, rise to the level for the programs that we have now that we have to require lumbar punctures. And there are other things that we can measure in CSF. In blood, there are many other proteins and biomarkers that we could potentially measure. I'm going to put that on hold for a moment because I want to turn to urine. Urine is actually not a biofluid. When I started thinking about biofluids, it's not one that I really prioritized high. There is some data looking at urine that says that we can identify a receptor from neurons in the urine. And it's called P75. It's a receptor that's on the surface of neurons. It's expressed highly during development, and then it kind of goes away in later childhood and adulthood. And it's expressed again if a neuron becomes ill. And not only is it expressed just meaning we find it there on the surface of neurons, but it also gets cleaved off and sent into the blood and the spinal fluid, and then cleared out in the urine. And we can measure the urine, and we can look at the levels of this. And what the previous studies have shown is that there is an increase in this P75 marker in urine over time in people with ALS. So we're applying that sort of still fairly preliminary evidence to the platform trial and measuring P75 in urine in at least one of the regimens. We do not have quite as much evidence supporting P75 or understanding how to interpret it or how much variability there is as we do, say, for neurofilaments, which have been studied much more extensively. Still really exciting to have these leads. I'm going to turn for a second to talk about speech analysis and home spirometry. And home spirometry, I think Jeremy's been on to talk, Jeremy Scheffner, that is, has been on to talk about home spirometry. The idea is really that home spirometry works very much like spirometry that we do in the clinic. That is another word for vital capacity. And we can do vital capacity at home, and we can do that very quite reliably, we think. We're really incorporating that into the trial to see if our hypothesis about that is true. If so, we can get more measurements at home, get them more frequently, reduce the variability that we have to deal with in the trial, and maybe get a better signal than just the ones that we do in the clinic. And that's why we're doing this. And it could be really important. I mean, especially Jeremy Scheffner has done a lot of calculations to show that if we have more measurements, it really can increase our ability to tell whether a drug is working. Speech analysis is something that we've worked on extensively in a number of different venues. So we've worked on it in a exploratory way to really understand speech with collaborators here and in Canada and across the US for some time. In the platform trial, we're using speech analysis with a company called Oral Analytics, who has really honed these algorithms and has kind of created a number of variables that we can come away with speech analysis and analyze. That is to say, when we make a recording of somebody speaking into a high quality recorder, there are some very simple things that we can analyze that tell us about disease progression. And those have to do, probably the lowest hanging fruit, that is to say, the easiest things to analyze from a speech recording are speaking rate, which we measure in words per minute, and articulation rate, which we measure in syllables per second. And we can see that those actually change over time in people living with ALS. There are other much more sophisticated analyses that we can do that either combine variables or rely on more acoustic analysis. So kind of what does the waveform of the sound look like? And how does that change? And do we see a change in the tone or the pitch or the kind of fluctuation in the waves over time? So we can analyze those things, and then we can combine those with some of the more basic measures like speaking rate and get new information. So we have about 8 variables that will be coming back from oral analytics from the speech recordings that we're making. Now, this has really not been used in a trial in this way for ALS. And so it's a really exciting time to have a lot of data come in and be able to really understand speech analysis as a metric and potentially as an important secondary outcome measure. And I would say that in conversations with regulators, as well as with industry partners and with academics, I think there's a huge will to really make this, to really explore this and understand whether it could be a key secondary endpoint for our trials. Right now, it really is an exploratory endpoint, but we're moving the needle forward as we rely on it to tell us about what's going on in these trials. And we're running out of time, but just lastly, I'll talk a little bit about DNA. So DNA is, there are lots of words for this: DNA, genetics, genomics. They all, have a similar meaning. That is to say that we can characterize the DNA, we can sequence the DNA from people living with ALS who are in this study. And then there are a number of things that we can do. Number one, we can identify any known causative ALS genes, and that will help us kind of categorize whether there are some genetic subtypes of the disease that may respond differently to an intervention. We also have the possibility of getting the entire sequence of the DNA, which would allow us to look for other variability from person to person, like risk genes, which we're beginning to understand in ALS. We're just beginning to kind of turn that corner. Or what we call pharmacogenomics. And this is sort of, we all process medications a little differently in our body. And some of that comes directly from our DNA and different sort of enzymes, different proteins that we have in our body to process drugs. And so increasingly, we're able to do analyses of the DNA and understand whether someone might be a rapid metabolizer of the drug. And so, if someone's a rapid metabolizer of a certain drug, they had a lower exposure to that drug, and that might inform us about why they didn't have as good a response. So that is, we're really looking for that to sort of come together and sort of help us lead the way for how we might incorporate these more off the beaten path uses of DNA.
Great, thank you so much, James. And not surprisingly, there's a number of questions about biomarkers, so perhaps I can ask them to you. I can ask them right now. They're very relevant to what you just described. So, first question: do you share neurofilament level results with participants of trials?
So this is a really good question. The answer to this right now is that we have not set this up to do that. And there are a number of reasons for that. The first one is that when we started the platform trial, there were no clinically certified tests for neurofilament. And we couldn't really return a research result. There are now some clinically certified results for neurofilament. The second reason that we don't return those results is that we actually don't test them until much later. We do them in batches. And the reason for this is because, we have to do them in batches. The reason we do them in bigger batches for our trial rather than smaller batches along the way is that we can cut down a little bit on the variability. So if we test a batch of 80 samples today, and a batch of 80 samples 4 months from now, there could be more variability than if we test 160 today. There are ways for us to overcome this, and we have put a lot of work into that, and we have what we call quality control samples and external quality controls, and we put a huge amount of work into that. So, but that's functionally why we've set this up this way.
Yeah. So are biomarkers included in futility checks for the different regimens?
So the futility, we have not incorporated biomarkers in those futility checks because they're still exploratory endpoints. So we wouldn't want to sort of declare a drug not effective because it didn't change one of these biomarkers, which are, most of which are still quite experimental. Neurofilament is probably our closest one, but as I said earlier, there's still some broad uncertainty about whether a drug has to change neurofilament in order to be effective. We wouldn't want to abandon a drug early just because it didn't have an effect.
Right. Along these lines, there's a question about the role of biomarkers in FDA reviews. So there's a few questions. One question is, do you think that there are any biomarkers that are ready for prime time? Are any biomarkers ready to be used to support accelerated drug approval? Or is it too soon for the FDA to use biomarkers?
I mean, I think you could, I think this is a really, really great question. And it's one that I think could generate a lot of discussion across the community, across every stakeholder in our ALS community, and I think we ought to begin having these conversations in earnest, and we are. My sense is that we're beginning to see, especially from the tofersen study results, the VALOR study results, that at least in the case of tofersen, there is a robust reduction in neurofilament, and that did seem to precede clinical effect, which is kind of how we thought this could work, is that you see a biological effect more rapidly, and then you see a clinical effect. So that is, I think, a shining example of the way that this could work. But I'm not sure that we can be, I guess the question is, can we be sure that if neurofilament goes down, clinical improvement or clinical stability or improvement or slowing of disease would follow? And we could have a discussion about that, and perhaps, but I'm not 100% sure of that yet myself. The next question is, if we don't see a reduction in neurofilament, can we be sure that there is not going to be a clinical effect? And I feel very uncertain about that myself. And I think, so I think, there may be scenarios where it is, where we're beginning to turn the corner. It'd be nice to have a second program that showed us both a reduction in neurofilament and a clinical benefit, to help us understand that if we see a neurofilament reduction, that we can expect that there'll be some clinical improvement. I think we may turn that corner before we are able to say, no change in neurofilament means no benefit.
There's a number of questions about very specific biomarkers, so perhaps we can take them like as rapid fire. And before we get to those, I just wanted to mention that, and as we see from the number of questions that we're getting, we've been receiving really many questions about biomarkers pretty much every week. And that's why we wanted to focus one webinar on biomarkers. And so maybe before we go to this rapid fire of different biomarkers that people have questions about, could you kind of summarize why are biomarkers, or why could biomarkers be important for people with ALS?
Yeah, thank you for backing me up a little bit. This is great. So what we'd like to do is run, number one, we'd love to have a test that we can do, that we can run that test, the blood test, and we say someone has ALS or doesn't have ALS. That's one use for a biomarker. We do this with diabetes. We can test glucose or hemoglobin A1C. It's a blood test, and you say that person has diabetes or doesn't. Maybe you have to do it twice, but there's really no question. We would love to be able to do that for ALS and make a diagnosis faster and get people into trials faster and get them on effective therapy faster. But there are many other uses for biomarkers as well. So here I've been mostly focusing on how do we use a biomarker to run a faster trial. So the other reason that people who are living with ALS might care about this is because we would love to run a 3-month trial where we can use a blood test to say this drug works or doesn't work. And a biomarker is something we measure in blood. Say we can measure it in other things, but we measure it in blood. We say, oh, this level has gone down, therefore this drug is affecting the disease. And so we don't need to study it any further. Our 3 months is effective, and we'll move on to approval.
Thank you. And so basically, with that background in mind, there's again a number of questions about the list of biomarkers. So perhaps really quickly, we can just share what they are. So somebody was asking about, many of these are still in development, as you just said. So can you comment on ERVK and HML2 as potential markers?
Yeah, so I mean, I think they're both in development. There are assays and there are champions for both of these. And I think that's kind of what it takes for us to really be able to move these, turn the corner from this is something that may or may not change with disease, to really understanding whether it does. And there are a number of community efforts to have biofluids on hand, have blood or spinal fluid on hand. The NEALS biorepository, Sabrina, you talked about earlier, and we house some of those samples here at Mass General. We share with researchers around the world, and we have shared them for both of these projects to really help us, understand whether these are potential biomarkers. They are potential biomarkers. Are they reliable in the way we need them to be? We're still trying to understand.
Right. What about neuronal conductivity? Is that a reliable biomarker, or what it is, I guess, to start?
Yeah, so there are a number. So what neurons are sort of like wires. They pass electricity, and it goes from, essentially it goes from the brain to the spinal cord. In this case, we'll talk about motor neurons, from the brain to the spinal cord, from the spinal cord out along the nerve, the muscle, and they're like a wire, and they carry electricity. And so we can measure that electricity, and we can measure it in a number of different ways, but we can measure it with just a sticky electrode over the skin and over the belly of a muscle, and we can stimulate over a nerve, and we can measure how much electricity that nerve is passing and compare that to what we think it should be passing. And we can do that in a lot of different ways. There are some techniques that we have used and that we can use. They tend to be a little bit variable in multi-center studies, and variable from one time point to another. So that variability has made it a little difficult. But there are really a cohort of people who are working on making better and better ways of looking at neuronal conductivity and making it useful in trials. And that work goes on, and I think there are some real hints that we are getting better and better at this.
Right. Another question, do you study structural variants?
So this is a complex question. Let me pull it apart a little bit. Proteins are formed and kind of take on a shape, a three-dimensional shape. And that shape may change if there's some illness going on that's affecting that protein. And so there actually are some real efforts now to look at critical shape changes in central proteins. So for example, there's a protein called TDP43, which is central to ALS. It's a protein that becomes sticky and sort of clumped together in the cells, in motor neurons particularly. And so TDP43 is there always, but when it's clumped together, that's a bad thing. So there are a number of programs looking at whether we can detect clumps of protein, clumps of TDP43, as opposed to sort of unclumped TDP43. And that's kind of a structural variant. Another way that we actually look at structural variants is in DNA. There is a test called EpiSwitch, which also uses some NEALS biorepository samples to look at its ability to predict fast versus slow progressors based on the shape of DNA.
Great. Last question about biomarkers. When you look at people with bulbar onset ALS and spinal onset ALS, any specific considerations in terms of neurofilament levels?
So neurofilament tracks better with rapidity of disease progression. There are a couple of studies that say that it's different in limb versus bulbar. I think we probably need to look more carefully at what stage of disease and how many regions are affected and so forth to really answer that question. Interestingly, so there's Answer ALS is a large observational study that we ran that has had over 1,000 people in it and allows us to look for subsets of participants both clinically and with biomarkers. And you would think it would be very obvious that we would find a biomarker that says these people are bulbar onset and these people are limb onset, living with the disease. And that has been harder than I think I would have predicted.
Right. So I know we have actually one question that was pending from last week. We promised that we would give the reply this week. So if you don't mind, I'm going to switch gears and ask our patient navigator team to answer that question. There was a question last week about people wanting to enroll in the New York, New Jersey, Connecticut area and which site was open. So Catherine, could you share? I know you looked into this, so if you could share the answer.
Yeah, sure. So hopefully that person is on the line tonight, but just in general, anyone is welcome to reach out to patient navigation with these types of questions. But in terms of the New York, New Jersey, Connecticut area, when I speak to people in that region, at the moment, the Hospital for Special Care in New Britain, Connecticut is the only site actively enrolling for Regimen E in that area. A couple other nearby options, relatively nearby, would be UMass Worcester in Massachusetts or Thomas Jefferson University in Philadelphia. So people in New Jersey, Philly might be within reach. If somebody's in Connecticut, the UMass might be another option. Coming down the pipeline, kind of forward looking, we're hoping to get Stony Brook University in Long Island up and running for Regimen E, hopefully by the end of this month, but the timeline could change. And then we're also in discussion with Columbia University, so more to come on that.
Great, thank you so much. And I know I just wanted to maybe quickly go through the slides just to make sure that we provide the weekly updates. I know people have asked us to continue to look into these. So we are still working on closing out the first 4 regimens to give results over the next few weeks or months. And then next slide, we are obviously enrolling for Regimen E again. And Catherine, thank you for sharing some site information. And these are the enrollment updates as of this week. Next slide. And these are the sites that are open again to follow up on that topic of which sites are available. We have 38 at this time, but we're working hard to open more sites. Next slide. And again, as always, if you have any questions, please contact Catherine and Alison, as you can see on the slide. And I think that was the last one. And before we wrap up, I just wanted to give you, James, the opportunity to say just a few more words about how the platform trial is contributing to biomarkers and why are we doing this, I guess. I know that the biomarkers are a hot topic in the community, so we want to contribute to that as platform group. So please tell us more about it.
Yeah, sure, absolutely. So I think there are 2 major reasons that biomarkers have, maybe 3 major reasons that biomarkers have such an important role in the platform trial. The first one that comes to mind is that we're trying to develop drugs and find new therapies for ALS. And biomarkers may help us do that for the specific drugs that we're testing in the platform trial. That's really the first reason that we include biomarkers. The second reason that we're including biomarkers and collecting biofluids that we're storing longer term and sharing with researchers is because we have the opportunity and the support of people with ALS who are participating in this trial and in these regimens to contribute to existing stores of biofluids that new research programs rely heavily on to kind of develop their molecules and get them faster to trial. And the third, which is kind of a sort of related to that one, is that we have some existing repositories or banks of blood and spinal fluid that we share with researchers doing biomarker research. But it is very rare to have these biofluids and potential sources of biomarkers in the setting of a clinical trial, where we can sort of match up any clinical effect and a biomarker effect. And that could be really transformative for how we understand biomarkers impacting future programs as well. So there's kind of we're doing it for right now, we're doing it for the greater good, and we're doing it really, really importantly for hastening drug development.
Thank you. And thank you so much, James, for joining us. Thanks, everyone, every panelist and the audience. And we'll see you again next week. Same place, same time. Thank you.
Great to see everyone. Thank you for joining. Bye-bye. Thanks for having me.